Hello everybody, this is Mona from BioClues presenting you a poster on laccases on behalf of all the authors. Laccases are enzymes produced by many fungi, plants and bacteria. These oxidoreductases typically contain three types of copper, of which one gives a characteristic blue color called the type 1 and the others lacking this are called the yellow or the white laccases. Generally, a minimum of 4 copper atoms are distributed over 3 binding sites per active protein for a catalytic activity of the enzyme. These copper binding regions contain 1 cysteine and 10 histidine molecules or amino acids, and this is strongly conserved in all the laccases. Laccases have several they are involved in several biochemical processes like lignin biodegradation, bioremediation, etc. Fungal laccases, due to their ability to break xenobiotic compounds using molecular oxygen and because of their role in psychological processes like pathogenesis, morphogenesis, pigmentation, detoxification, etc., find application in the food, paper, textile, pharmaceuticals, and wastewater management industries, and also in nanobiotechnology. So, because of these reasons, earlier one of our team, including Shiv, who's also an author to this project, performed delignification studies on about 10 white rot fungi using various agricultural residues and found Pycnoporus species of the Polyperaceae family to show maximum delignification activity when compared to the rest, which is considered to be the maximum rate reported for any fungi. So, of late, we decided to further continue our research on laccases and we performed some bioinformatic analysis to understand the domains of these laccases and their conservation through evolution. The NCBI search gave us 1,285 fungal laccases, of which only 73 were for Pycnoporus. So we performed domain search using ProCite, ProDom, PFAM, and NCBI conserved domain search to reach consensus and found either or all the three copper oxidases, that is copper oxidase 2, and copper oxidase 3, were present in all the laccases. Next, we compared the random sequences from various organisms using UniProt to see if we are able to find any common conserved region in the organism or across various organisms. However, we couldn't reach any similarity, any consensus. So, narrowing down the search, we aligned sequences only from fungi of different family, and still no conserved regions were found. Finally, when the alignment was made specific to Polyperaceae family, a high level of similarity and matches were found, suggesting that the function and evolutionary conservation of domains are varied across different levels of organisms when compared to a particular family. When we analyze the result in case 3, we identified polar amino acids, serine, threonine, were preserved to a specific position or specific positions at various stretches. Now, polar amino acids are prone to be formed at the active sites and are involved in substrate binding. And thus, this result indicates that the laccases have high enzymatic activity. Now, cytochrome C, ubiquitin, cytochrome P450, and laccases, they all share similar functional involvement in hydroxylation reactions in lignin and aromatic compound degradation. So we performed a protein-protein interaction study using gene mania and osprey to see if there are any links between these molecules. And under human grid only, we found that cytochrome C is co-expressing with laccase, and so do cytochrome P450 through a two-step interaction, that is with through CLK1 and SIRT4. However, we couldn't find any link to ubiquitin and between ubiquitin and laccase. So, based upon this result, we believe that further understanding the relationship between all these molecules would be interesting, and this could be exploited in the lab, and we could introduce these molecules externally in the medium to induce laccase production. So, thank you so much for your time and for listening to this presentation. Have a good day. Thank you.